consists of those microorganisms which are present on body surfaces, covered by epithelial cells and are exposed to external environment, and are present in GIT, vagina, and respiratory tract. The number of bacteria colonizing mucosal and skin surfaces exceeds the number of cells forming the human body. The most abundant microflora is present in the distal parts of the gut. The majority of the intestinal bacteria are gram-negative anaerobes. The examples of commensal bacteria are E. coli in gut, Streptococcus salivarius in oral cavity, Corynebacterium acculensis in skin and nasal cavity. Uses. Actinobacteria produce secondary metabolites that have antibiotic, antifungal, and anti-cancer properties. Streptococcus oralis can induce protection against miliary infection, referred commonly as otitis media. E. coli helps the body break down and digest the food. Lactobacilli can help treat diarrhea, vaginal infection, and skin disorders like eczema.
Pathological bacteria in gut microbes refer to special bacteria which causes infection in gut microbes. Lot of gut microbes are beneficial, but some special strains causes infection in our human body. So first example of this is Escherichia coli. The special strain which causes infection is E0157S7, which causes food poisoning, diarrhea, abdominal pain. Sometimes it can be life-threatening. Second one is Salmonella species, which causes food poisoning. They can contaminate various foods like street foods, raw egg, poultry, etc. Due to that food poisoning.
Recent studies have elucidated substantial divergences in the microbiome structure between healthy individuals from different race and ethnicity. Some of the geographical or racial variations in microbiome structure have been attributed to differences in host genetics and innate immunity, while in many other cases, cultural, behavioral features like diet, hygiene, parasitic load, environmental exposure, etc. overshadow genetics. Between group differences in susceptibilities to many health conditions, from preterm birth to type 2 diabetes, obesity, and even cancer are being linked to microbiome diversity. I'm going to list out a few examples. Native Africans have a high dietary intake of animal proteins and fat, but a low fiber intake by African Americans than Native Americans. In Native Americans, there's a high dominance of Prevotella and butyrate producing groups. In European children, they typically have a Western diet high in animal protein, sugar, starch, and fat, and low in fiber. Due to this, they have a high abundance of firmicutes, proteobacteria, and enterobacteria.
Meiosis of the gut bacteria communities in patients or animal models may cause many diseases. For example, antibiotic treatment and surgery cause pseudomembranous colitis due to toxin production by Clostridium difficile, and sepsis of Escherichia coli, Enterococcus fecalis, and intra-abdominal abscesses due to Bacteroides fragilis. Imbalance in gut bacteria composition was associated with intestinal symptoms such as bloating, abdominal pain, and diarrhea. Uncultured phenotypes from Clostridium clusters 4 and 14A have statistically significant positive correlation with bloating. Inflammatory bowel disease (IBD) is most common in the developed countries of Europe, the US, and Scandinavia. Reciprocal interaction between commensal gut bacteria and the host may induce allergies and IBD. Overly aggressive Th1-mediated cytokine response to commensal bacteria may be the pathogen of chronic intestinal inflammation. In addition, disorders in bacterial recognition by macrophages are strongly related to pathogenesis of IBD. Furthermore, IBD could result from an abnormal immune response against the commensal microbiota in a genetically susceptible host. Crohn's disease is another type of inflammatory bowel diseases. It has been thought to be an autoimmune disease in which the body's immune system attacks the gastrointestinal tract and causes inflammation. Various gut microbial metabolites such as SCFAs, TMAO, and tryptophan-derived metabolites have been reported to be closely related to the pathogenesis of type 2 diabetes mellitus. SCFAs inhibited insulin signaling by activating G-protein coupled receptor 43 in adipocytes, thereby suppressing fat accumulation in adipose tissue and promoting energy metabolism in other tissues. Probiotics are made of good bacteria that helps keep your body healthy and working well. This good bacteria helps you in many ways, including fighting off bad bacteria when you have too much of it and helping you feel better. You have trillions of microbes on and in your body. These microbes are a combination of bacteria, fungi, viruses, and protozoa. The most common place linked to beneficial microbes is your gut. There are two specific types of bacteria that are common probiotics found in stores. These include Lactobacillus and Bifidobacterium. Probiotics are also made of good yeast. The most common type of yeast found in probiotics is Saccharomyces boulardii. Effects of probiotics. Probiotics are live microorganisms. When taken in sufficient amounts, they can help restore the natural balance of gut bacteria. They can reduce the risk and severity of diarrhea from a number of different causes. Research shows taking probiotics may help improve symptoms of mental health, such as depression, anxiety, stress, and memory. It protects the heart from reducing bad cholesterol and modestly lowering blood pressure. They may reduce the risk and severity of certain allergens. Probiotics may help reduce the symptoms of bowel disorders like ulcerative colitis, IBS, and necrotizing enterocolitis. It also helps boost your immune system and protect you against infection. Some people may experience headaches after using these food and should opt for probiotic supplements. Immunocompromised people, venous catheters, recent surgery, acute pancreatitis, or prolonged hospitalizations should avoid taking
In pharmacogenomics, genomic information is used to study individual responses to drugs. When a gene variant is associated with a particular drug response in a patient, there is the potential for making clinical decisions based on genetics by adjusting the dosage or choosing a different drug, for example. Scientists assess gene variants affecting an individual's drug response the same way they assess gene variants associated with diseases, by identifying the genetic key associated with known drug responses and then testing individuals whose responses are known. A common CYP450 enzyme, for instance, is CYP2D6, which affects how the body processes several medications such as codeine. Some individuals have increased CYP2D6 enzyme activity and break down codeine so quickly that a standard dose can lead to dangerous side effects. Others have decreased CYP2D6 enzyme activity and do not activate codeine to enable it to work, and so they may not get pain relief when taking codeine.
Host genetics and the gut microbiome can both influence metabolic phenotypes. However, whether host genetic variation shapes the gut microbiome and interacts with it to affect host phenotype is unclear. The gut microbiome is environmentally acquired from birth, therefore it may function as an environmental factor that interacts with host genetics to shape phenotype, as well as to genetically determine attribute that is shaped by and interacts with the host. For instance, the concordance rate for carriage of the Methanobrevibacter smithii is higher for monozygotic than dizygotic twin pairs.
Phage therapy, also called bacteriophage therapy, uses viruses to treat bacterial infections. They are harmless to people, animals, and plants. The word bacteriophage means bacteria eater. They are found in soil, sewage, water, and other places. This therapy for disease-causing bacteria may be a useful alternative to antibiotics. Bacteriophages kill bacteria by making them burst or lyse. A virus infects the bacteria by injecting its gene, which is the DNA or the RNA. The phage virus copies itself and reproduces inside the bacteria. Finally, the virus breaks open the bacteria, releasing the new bacteriophages. Just as there are many kinds of bacteria, there are several types of bacteriophages. But each kind of phage will only attack a certain bacterium and won't infect the other kinds of bacteria. Drawbacks of phage therapy include that the phages are currently difficult to prepare for use in people and animals. Phages may also trigger the immune system or overreact and cause an imbalance.
If you are experiencing recurring infection with Clostridium difficile in your colon, your doctor may recommend a fecal transplant. It is a procedure to collect feces from a healthy donor and introduce them into a patient's gastrointestinal tract by adding healthy bacteria into the recipient's intestine. FMT can be performed in children and adults.
It is usually performed by colonoscopy. A gastroenterologist guides a colonoscope through the entire length of the colon, and as it is withdrawn, the solution containing donor feces is deposited into the colon. But you might experience temporary side effects such as bloating and intestinal gas, cramping due to the air trapped in the colon during the procedure, constipation due to anti-diarrhea medication.